Hey Olivia, how you doing?
Good. How are you, Laura?
Good. It's so funny running into you here on this Zoom call. I
I
just
know.
was
I can't believe it.
just by chance alone, and I just was wondering. I'm looking for somebody to teach me a little bit about a PCOA, why I might use it, and how I might do that. And I'm hoping to do that in Karime. Write it down. 12 minutes. Last time I said 10 minutes. It took 20 minutes. What do you
Mhm.
think, Olivia?
I think I can handle that. I might actually have some slides ready to go just for this reason.
You have slides?
I have
Wow, cool.
slides.
Okay,
Uh,
I'll be quiet. I'm going to listen and learn.
let's see if I can share. Can you see my screen?
Absolutely.
Perfect. Okay, so I'm gonna talk about a little bit about my honors project so that I can sort of set the scene for why I use PCOAs for my project, which will relate to the 371 stuff just because it's so similar. But basically, my project is looking at test state amoeba communities and specifically their diversity across space and time in New England bogs. Can I change the slides? Yes. Um, so a little background on test state amoeba for people who might not know is that they are a unicellular eukaryote or a protist. They build shells. They're in the order Arcelonida, and their shells can have a variety of shapes, as we can see in these images down here. Um, we care about test state amoeba because they're important bioindicators in sensitive environments, and their populations can be analyzed to determine the health of their environments because they're so sensitive to environmental factors like pH and temperature. So the goals of my project are to combine molecular and morphological techniques to get a clear image of these communities and to look for patterns over time and space. These are some images from places we sample. My methods are basically the same as the methods from lab. So we went sampling, we did sample preparation. Then on the genetic side of things, we did a DNA and RNA extraction of the community, a PCR, and then Illumina high throughput sequencing into our data analysis. And I just have the added portion of a morphological aspect. We did sampling all across New England in different bogs in Massachusetts and then some bogs in Maine. We measured pH and temperature of the water that they were living in. We sampled from March through October in 2018. And we just want to know how these communities change through seasons. Basically, are the bogs that are closer, are those communities more similar to each other? And are the bogs homogeneous? So are there differences within space in an individual bog? But for this exercise, we're just going to focus on holly bog. So these are some images I put together from a time we went sampling back in 2018. And you can see in this left image that we have this measuring tape, and we would take samples about every 5 meters or so, going from the forest line out towards the open bog, which you can see over here. And it's really cool. They've got these boardwalks. And so my hypotheses are that the tested amoeba communities will be more similar the closer in space that they are, and that they'll be different within the bogs due to environmental differences across space, and that the communities will be dynamic over time as seasons change. So the thing that I thought would be best to use, let's see if I can share my other screen, new share, in our studio. So the way that we can look and see how similar or different these communities are from each other is to make a PCOA, or a principal coordinates analysis. And so I'm going to walk you through the script that I developed with Naomi Ostriker, who's also in 371, for our bioinformatics class last semester. And it's been a very tried and true script that I'm very, very pleased with. So the first thing that you need to do always is load your required packages. Actually, let me sweep my environment so we can see it all happen at once. There we go. Let me move over here. So we're going to load the required packages and then load the data. So the nice thing that we learned is that you can put a CSV into Google Drive and then have R read that CSV from Google Drive. So you can access these files from any computer without having to put in a specific path name. The only file that requires a specific path name is the tree file, which is held on my desktop for this purpose, just to make the path name easy, but that'll be specific to the computer that you're using. So once you load these in, you should be able to open it, and you'll see that it's an OTU table very similar to the ones, or basically exactly the same to the ones that we use in class. So you have your OTUs, all of these blast data and taxonomy data, and then the more that you scroll over, then you start to get into the samples over here. The sampling sheet is something that you'll make for your data. We found this was just a really easy way to keep everything organized. So it has all of your sampling sites, what they're called, where they're located, when they were sampled, whether they're DNA or RNA. This was just a test thing that I did to make sure it was working. And then for Holly specifically, we broke them down into areas of middle, open, and forest in relation to like what part of the bog that they were in, whether they were closer to the trees or closer to the open bog. So we can just look at those, make sure that everything's loading properly. That's just telling you that everything is okay. So the first thing that we need to do is take the reference sequences out of our tree. So this is just some scripts to drop those reference sequences out of our tree, so we're left with a tree that has only OTUs in it. Then we want to get rid of the rare OTUs, and rather than doing this by hand, we just decided to have R do it. So it takes any OTU with less than 100 reads and it removes it from our sampling or from our OTU table. Then I want to make a data frame of those OTUs that we've already filtered that are just from Holly Bog. So we're removing any samples that start with A for Acadia National Park, F for Harbor Forest, Orono Bog, and Bear Swamp. So then when we do that, we should get this thing called filtered OTUs Holly, which is I think here. And now when we scroll over to see our samples, there should only be samples that start with H's. So that's just a good way to check and make sure that everything is working correctly. Why is it in my way? I don't know. I'm gonna take, I don't know what happened with this, but now I can't access my thing. Oh, there we go, okay. So then, once we've done that, this creates our sampling info data frame. So that thing that we saw earlier that had just the samples and then the information that was relevant to what type of samples those were, we want to make sure that that's also only got Holly Bog in it, because it won't work if there's a different number of samples in the info sheet and the OTU table. Then a lot of this stuff is just so that we can get these matrices that match all of the data together so that the script can run properly. So it creates all of these weird new files and then matches them back together. Then we need to get these matrices to match their respective classes. So basically we said, okay, the location of our samples is you're going to take it out of this sampling info sheet and it's going to be the column titled sampling location. So this is just to get us some like more useful names for all of our information about our sampling sites. So then we run that and we have our data frame, and then we have to merge our data frame and get everything ready to go. This is just a bunch of like, this is basically written into the code for PhyloSeq, which is the program that we're using, or the package that we're using in R. So once everything is set and all of the files are matched up with one another, everything's merged together properly, then we can finally make our plot. So there's a few different things that you can put into, or that you can, a few different factors you can add into your plot. So we're going to use the method PCOA because we're doing a principal coordinates analysis. When we say that the distance is unifrac, that means that we're taking into account how related OTUs are to one another based on the tree that we made. And if we weight it, that means that rare taxa will not be taken into account with the same, I guess weight is the best word to use, as taxa with a lot of reads. So that way we can't get our samples skewed by just a few not very abundant OTUs. So then we're going to plot our PCOA. I'm going to have it make the color be for the month sampled and the shape be for the sample type, so whether it's DNA or RNA. And when it runs, it looks like this. And so we come out with this very cool looking plot, very colorful looking plot. So if we want to see a little better how each month looks on its own, we can use this function called facet wrap. And when we facet wrap across the month sampled, then we get these nice little boxes that are individually split up by month. And you can basically put in like any, let's say that I want to look at the, what's it called again? The area rather than the month sampled in the bog. Now the color is split up by whether it's in the forest, the middle, or the open bog. And so we can see that the forest is kind of all concentrated right here, but the middle and the open bog are basically spread out across the whole graph. So you can play around.
Maybe I saw one other cool thing on that graph too. I see that the forest is always clustered, whereas the middle and the open vary, but I also see that your DNA, which are the circles, seem to occupy a different space in the blue and green color than the RNAs, which are the triangles. I'm also amazed in this particular version of your data that 60% of the variability in your data set is being explained on that x-axis and 20% on the y-axis. So you're explaining the bulk of the variance in this two-dimensional graph. So I see forest, everybody else, and I see middle open DNA being really distinct. So the present community in the middle of the bog and the open bog are, is that open water? I see in the middle of the bog and near the open water are quite distinct from the RNAs.
Yeah, it's really, and then when you look at it with the facet wrap on too, it's like even more apparent that the forest, like the DNA and RNA are all very similar to one another. And in the middle, you've got this like really interesting cluster of DNA all the way over here. And then you're seeing the same thing out in the open bog. So I'm very excited about this, actually. I just discovered this today. So this is going to be fun for me to look into. But yeah, so that's basically how the script works. There's a lot of like behind the scenes computer magic.
Unsharing your screen, we can talk for maybe one more minute.
Yes.
Good. Good. So maybe you can say another word too, and maybe you could say just in a few words what you expect those people from 371 to take away and where you're prepared to help when they try to run their own scripts.
Yes. So hopefully what you guys can take away from this is that PCOAs are a really powerful tool for being able to visualize your data in a way that takes into account the different kinds of samples that you took, and you can really look at a lot of different details. So you could split your pitcher plants by age and split them by DNA type. You could split them by species. So as long as you have that sampling info ready to go, you can look at all kinds of crazy patterns in your data. And as far as things that I'm able to help with, I'm definitely able to help you guys get your scripts set up to be successful with your data. We can meet in small groups, and I can help you guys get your sampling info sheets set up and different things like that. You definitely do not need to take away from this how a PCOA is computed or how the script, you don't need to be able to write the script on your own and do it on your own, but we just want you to see it so that way you're ready when we are ready to dive into your own data.
Beautiful. And so this work we're going to do from the week of April 7th. So people might watch the video, this video, hopefully before April 7th, but don't worry about it because I think you said that you might be available for one-on-one group appointments, one-on-one or group appointments for the from the 7th to when we'll have something due on the 14th.
Definitely.
Well, do you have a last word you want to say?
don't think so. I hope you guys enjoyed the video. I hope that it made sense. That's it.
I thought it was really lovely. I learned a lot, actually. So thank you, Olivia. And it was just so nice to run into you, happen to pass
Yes.
by you. So take
You
care,
too.
Olivia, and take care, everybody else. So.